Okay, so as you guys have highlighted that you need a little bit of help with DNA structure and function, I'm going to go through that with you now in this little video. Okay, so we know that we're looking at DNA. Okay, and DNA is the chemical which carries the genetic code. So it carries the genetic code and it tells our body how to make protein. Essentially, that is the job of DNA. Okay, we're going to look at how that works in a little more detail after we've looked at the structure of DNA at a molecular level. Okay, so DNA is a polymer. Which means it is a very large molecule which is made up of repeating units or monomers. Okay, so we're going to see the same structures repeated over and over and over again in DNA to form its double helix. So we know on a slightly larger scale that DNA forms a double helix. Okay, excuse me, double helix. Okay, a double helix means that it is made from two different strands. Here is one strand, and here is strand number 2. Okay, those two strands are twisted around each other. Okay, so we've got two strands here, twisted around each other to form this double helix here. Okay, so that's the very recognizable, easily recognizable form of DNA that we've all seen in lots of different images. Okay, so let's try and zoom in a little bit on DNA and what its structure is on the more chemical level. Okay, so we've said that DNA is made up of monomers, and those monomers are called nucleotides. Okay, and those nucleotides have a very specific structure, and they are repeated time and time again inside DNA to make the DNA molecule. So a nucleotide is made of 3 different chemicals. So first of all you have a phosphate group, so here is phosphate. And attached to the phosphate you have a sugar, which looks a bit like that. So here is a sugar, and attached to the sugar you have one of 4 different bases. Okay, so that particular structure right there is what we would call a nucleotide. See if we can just pick up this nucleotide and take it with us. Okay. Okay, so here's our nucleotide. Let's make it a little bit smaller. Move it over here. Okay. So these nucleotides then can be repeated time and time again. So we have another phosphate here, which joins to the sugar, and then that phosphate joins to the sugar on the same nucleotide, and then here we also have a base. Okay, so again we've got sugar, phosphate, base. Okay, now we've got 2 areas that we need to look at here. So we have the beginnings of what is known as the sugar phosphate backbone. Now if we're to draw the second strand of DNA, we have to draw it in the opposite direction. So we'll start down here with a phosphate group joined to a sugar. Notice how the sugar is upside down now. And oops, I have to draw it a little bit differently to get the base in. The two bases need to be lined up with each other because they're going to join to each other here with hydrogen bonds, okay? And then we repeat down here again. So here we've got another phosphate joined to another sugar, and then the base from that particular nucleotide bonding to the base from the other nucleotide, okay? So what we can see from this is they're upside down, they're in opposite directions, and they are read in opposite directions too. So let's have a little look, a little closer look at what's happening here with these bases, okay, that are joined together. And the bases, there are 4 of them, are represented by 4 different letters. So you can have an A, a T, a G, or a C in DNA. And A always joins with T and G always joins with C. Okay, that's the only way they can work. So if we have an A here, over here we have a T. Okay? Similarly, if we have a G on this strand, then we have a C on this strand. Well, don't forget that you can change that around. So instead of having a G on this side, you could have a C here, which would mean G on the other side, for example. Okay, the way I remember this is AT at the GC Golf Club. Okay, so now we're starting to look at how the code works in DNA, and now we can start to think about, well, what does this mean in terms of genes? Okay, so if we start to look at what this could mean in terms of genes, okay? So a gene, what is a gene? Okay, a gene is a section of DNA, or a section, a sequence of bases. And what does that section of DNA or sequence of bases do? It codes for one protein. Okay, how does it do that? So the code on DNA is read in a series of 3 bases. Okay, each one of these is called a codon. So a codon is 3 bases, and they code for one amino acid. Okay, you take lots of amino acids, multiply, put them together, and you make a protein. Okay, so let's have a look at an example of that. So you might have, for example, on a strand of DNA, you might have ATC. And then the next codon might be GAC, for example. On the other strand, obviously, you would have the complementary sequence, which in this case would be T, A, G, C, T, and then, G. Okay? Well, we don't need to worry about that too much at the moment. We can just remove that for now and focus on one strand. So when we read DNA, we look at this single strand, and the DNA is read in terms of its codon. So here is one codon, and here is another one. There's read in these 3-letter words called codons. Okay, so this one codon here will code for one amino acid, and this codon here will code for a different amino acid, here represented by the different colours. Okay, we join those amino acids together, and we've started to make a protein. Okay? Okay, but for amino acids, there are only 20 amino acids on Earth. And the possible combinations of 3 different nucleotides to make codons is 64. So we can see that we have many more codons than we do amino acids. Okay? And that means that you can have more than one codon for every amino acid. So as we showed before, one codon codes for an amino acid, but there could be several different codons that code for that same amino acid. Okay, so to just review the key terminology from this video, then I would say that you need to know the following. You need to know what a nucleotide is and its structure. Okay, so nucleotide. You also need to know what a double helix is. You need to know what a gene is, a codon, a protein, you need to know how that's a B, by the way, how the bases pair up. So you need to know about your base pairs. And I would also suggest that you look at the sugar phosphate backbone, and always remember to include that in your descriptions of the structure of DNA.